Today, we are going to talk about Ig class switching.
Today's topic is here.
First, we talk about the definition. B cells expressing membrane-bound IgM and IgD on the cell surface, mature B cells, are activated to proliferate and produce secretory IgM and IgD when they encounter a specific antigen to which they bind. These mature B cells then differentiate into antibody-producing cells. In this process, the production of secretory immunoglobulins increases and they produce IgG, IgE, IgA, and other antibodies in addition to IgM and IgD. This is called Ig class switching.
Let's take a deeper look at Ig class switching. Ig class switching is caused by class switch recombination of the immunoglobulin Ig constant region. Constant regions do not directly react with antigens but play an important role in immune system signaling.
Next, we talk about mention how the class switch occurred. First of all, the class of the antibody is determined by the Shi gene sequence in the heavy chain constant region. If the gene is encoded by the Shi mu gene, IgM is produced. And if the gene is encoded by the Shi delta gene, IgD is produced. IgG, IgE, and IgA are similarly produced by each Shi gene. Changes in the coding region of the Shi gene leads to the Ig class switching.
The heavy chain constant region has switch regions. The switch region plays an important role in the recombination initiation of the Shi genes. Key enzymes associated with the recombination process include activation-induced deaminase, AID, which specifically targets the switch region. This leads to nicks in both standards of the switch region on which AID is activated. Such nicks facilitate the recombination of the switch regions.
Based on the above, we mentioned how the class switch occurred, using the conversion of IgM to IgG as an example. This is the heavy chain gene sequence of a mature B cell expressing IgM. AID specifically acts on S microregion and S gamma 1 region of this sequence to make a nick. Then, DNA between the switch regions forms a loop and is cut out as non-functional circular DNA. As a result, the C gene moves into the vicinity of the recognized VDJ sequence, and the recombination has occurred. Finally, the gene is the heavy chain's locus of an IgG expressing B cell is formed and IgG is produced.
Finally, we talk about what is a biological role. The point here is that the barrier regions do not change between antibodies, so all antibodies produced by the Ig class switching bind to the same epitope. Therefore, antibodies with different functions are produced against specific antigens.
Thanks to Ig class switching, it is possible to eliminate antigens more efficiently. Before Ig class switching, only two types of antibodies attack the antigen. In this case, the immune responses are not bad. After Ig class switching, all class types of antibodies attack the antigen, and all kinds of immune responses are induced by the antigens. In this case, the immune responses are good. Therefore, the antibodies eliminate them efficiently.
The summary is here.
References. That's all. Thank you for watching this video.